A pistol shrimp collapsed its own brain. This is what happened to the ocean. Pinky is a 4-year-old male Alpheus heterocheilus, a pistol shrimp, living in the coral reefs off the coast of Indonesia. He's approximately 5 centimeters long, translucent with bands of orange and white across his carapace, and like all pistol shrimp, he possesses something extraordinary, a specialized claw capable of creating one of the loudest sounds in the ocean. For years, Pinky lived a normal life. He snapped his claw to stun prey, to communicate with his mate, to defend his territory in the reef's crevices. Each snap produced a cavitation bubble, a void in the water, that collapsed with such force it generated temperatures as hot as the surface of the sun for a nanosecond, producing a shockwave that could stun small fish. But then, something changed. Marine biologists first noticed the anomaly 3 months ago. Dr. Sarah Chen, a researcher from the University of California studying invertebrate neurobiology, was conducting routine acoustic monitoring in the reef when her equipment picked up something impossible. The frequency was wrong, she would later report in her preliminary findings. We were detecting snap sequences at 14 times the normal rate, and the amplitude, the volume, was increasing exponentially. Pinky was snapping continuously. Not for prey, not for defense, just snapping. Let me explain what makes this so dangerous. The pistol shrimp's claw operates through a mechanism called cavitation. Cavitation, from the Latin cavus, meaning hollow, the rapid formation and collapse of vapor cavities in a liquid. When Pinky snaps his claw shut at speeds exceeding 100 kilometers per hour, he creates a high-velocity water jet. This jet generates a low-pressure region, causing the water to literally vaporize for a microsecond, forming a bubble. When this bubble collapses, a process called implosion, it releases an enormous amount of energy. We're talking about temperatures reaching 4,700 degrees Celsius. That's nearly as hot as the sun's surface. The collapse also produces a flash of light called sonoluminescence. Sono, meaning sound, lumen, meaning light, light from sound. This is a weapon, a precisely controlled biological weapon that stuns prey in milliseconds. But weapons can misfire. By week 2 of the observation period, Pinky was snapping approximately 840 times per hour. Normal shrimp snap maybe 60 times. His major claw, the dactyl, showed signs of structural damage. Microfractures appeared in the chitin, but he didn't stop. Dr. Chen's team decided to intervene. They carefully captured Pinky and placed him in a specialized observation tank with acoustic dampening equipment. We needed to understand what was happening to his nervous system, Dr. Chen explained. This wasn't hunting behavior. This wasn't territoriality. This was something else entirely. They began running neurological scans. The pistol shrimp's nervous system, while primitive compared to vertebrates, is remarkably sophisticated for its size. The cerebral ganglia, what we might loosely call a brain, consists of approximately 100,000 neurons clustered near the esophagus. These neurons coordinate sensory input from the antennae, visual processing from the compound eyes, and motor control for the specialized claw mechanism. What the researchers found was unprecedented. Pinky's motor neurons, the neurons responsible for triggering the claw snap, were firing autonomously, continuously, without input from sensory neurons. It was as if the snap command had become stuck in an infinite loop. We were looking at what I can only describe as a neurological cascade failure, Dr. Chen said in her notes. But it got worse. By day 17 in the observation tank, Pinky's snap rate had increased to 1,200 per hour, 20 snaps per minute, one snap every 3 seconds. The acoustic energy in the small tank was creating what physicists call resonance amplification. Resonance, from the Latin resonare, to resound. When wave frequencies align, they amplify each other. The tank's dimensions were accidentally creating a standing wave pattern. Each snap wasn't just producing energy, it was reflecting back, intensifying, adding to the next snap's power. The water temperature in the tank increased by 2 degrees Celsius in 6 hours. Pinky was, effectively, cooking himself. But the most disturbing discovery came from the neurological data. Dr. Chen's team used a modified micro-CT scanner designed for small crustaceans to image Pinky's cerebral ganglia at high resolution. They found massive neuronal death, apoptosis, apo, meaning away from, ptosis, meaning falling, cells falling away from life. Programmed cell death. Except this wasn't programmed. It was being triggered by the shockwaves. Each time Pinky snapped his claw, the cavitation bubble's collapse sent shockwaves through the water. In the ocean, these dissipate harmlessly. But in the small tank, with the resonance amplification, these shockwaves were bouncing back through his own body, through his carapace, into his neural tissue. The pressure waves were literally shaking his neurons apart. This phenomenon is similar to what human divers experience with decompression sickness, or what soldiers experience with repeated blast exposure leading to traumatic brain injury. The mechanism is different, pressure versus vibration, but the outcome is the same. Neurons can't maintain their delicate electrochemical gradients when subjected to repeated mechanical stress. In Pinky's case, the stress was self-inflicted, continuous, escalating. Dr. Chen's team faced an ethical dilemma. They were observing a creature destroying its own brain, driven by a neurological malfunction they didn't fully understand and couldn't stop. On day 23, Pinky's snapping rate reached 1,680 per hour, 28 snaps per minute. The researchers could see visible tremors throughout his body between snaps. His antennae, normally active and exploratory, hung limp. His compound eyes, normally tracking movement, stared fixed in one direction. The cerebral ganglia showed 60% neuronal death on imaging. For context, humans experience severe cognitive impairment at just 10% neuronal loss in key brain regions. Then, on day 24, the snapping stopped. Pinky had gone completely still in the tank. Post-mortem examination revealed catastrophic neural degeneration. The cerebral ganglia had essentially liquefied, the neurons so damaged by continuous mechanical stress that the tissue could no longer maintain structural integrity. It was, in the most literal sense, a brain collapse. But here's what makes this case scientifically significant. Pinky wasn't just an anomaly, he was a warning. Dr. Chen's team went back to the original reef where Pinky was collected and conducted a broader survey. They found 23 other pistol shrimp exhibiting similar symptoms: elevated snap rates, erratic behavior, neurological stress markers. All of them were in areas of the reef experiencing human-caused acoustic pollution. The shipping lanes had recently been rerouted closer to the reef. The constant low-frequency thrumming of diesel engines, the sonar pings from commercial vessels, the pile driving from coastal construction, all of it was creating an underwater noise environment that was interfering with the pistol shrimp's ability to regulate their snap behavior. Marine biologists have known for years that anthropogenic noise affects marine mammals. Whales beach themselves, dolphins abandon feeding grounds. But this was different. This was noise causing an invertebrate to essentially enter a neurological failure state that resulted in self-destruction. The mechanism appears to be tied to how pistol shrimp use acoustic feedback to regulate their behavior. When they snap their claw, they listen to the result, processing the acoustic signature to determine if the snap was successful. This feedback loop helps them adjust snap force, frequency, and timing. But in a noise-polluted environment, this feedback system fails. The shrimp can't properly hear their own snaps above the background noise, so the motor neurons keep firing, trying to achieve the acoustic signature they're programmed to expect. It's like pressing an elevator button repeatedly when you don't hear the confirmation beep, except the shrimp can't stop pressing the button. Ever. The team published their findings in the journal Marine Ecology Progress Series 3 weeks ago. The paper is titled "Anthropogenic Acoustic Pollution Induces Fatal Neurological Cascade Failure in Alpheus heterocalis." The implications extend far beyond one species of shrimp. Pistol shrimp are keystone species in many coral reef ecosystems. They create burrows that provide shelter for other species. Their snapping creates acoustic complexity that many fish use for navigation. Their predation on small invertebrates helps control reef pest populations. If noise pollution is causing widespread neurological damage in pistol shrimp populations, we could be looking at a cascade failure not just in individual animals, but in entire reef ecosystems. And here's the deeper concern. If we're seeing this effect in an animal with a relatively simple nervous system, 100,000 neurons, what might be happening to creatures with more complex neural architecture who also depend on acoustic signaling? Dolphins? Whales? Deep-sea fish? Dr. Chen's team is now working with environmental agencies to establish noise reduction zones around critical reef habitats. Some shipping companies have voluntarily rerouted to avoid sensitive areas. The International Maritime Organization is considering new regulations on vessel noise emissions near marine protected areas. But the work is slow. The ocean is vast. The shipping industry is global. And the noise, for now, continues. Pinky died from a condition that, in medical terms, we might call acoustic-induced traumatic encephalopathy with catastrophic neurodegeneration. But more simply, he couldn't hear himself think, so he thought himself to death. His case has opened a new field of study in marine biology, invertebrate neurological responses to anthropogenic stressors. Researchers are now examining whether other species that rely on acoustic signaling might be experiencing similar effects. The findings could reshape how we think about ocean conservation. Because here's what we've learned. Evolution gives animals extraordinary abilities. The pistol shrimp's claw is a marvel of biological engineering, a tool for survival refined over millions of years. But evolution doesn't prepare species for the rapid changes humans introduce to their environment. Changes that happen not over millennia, but over decades, years, months. Pinky was 4 years old when he died. For a pistol shrimp, that's approaching middle age. He should have had years ahead of him, hunting, mating, defending his territory with those spectacular cavitation snaps. Instead, his own weapon became the instrument of his destruction, triggered by an environment his species never evolved to navigate. The ocean is not silent. It never was, but it used to be filled with the sounds of life, whale songs, dolphin clicks, the snapping chorus of shrimp that sounds like crackling fire from above the waves. Now, increasingly, it's filled with the sounds of us. And for the creatures who call it home, who navigate by sound, who communicate through acoustic signals refined over evolutionary timescales, our noise isn't just annoying, it's deadly. Pinky's story ended in a research tank in California, but the story he tells is still being written in oceans around the world, in coral reefs and deep-sea trenches, wherever our noise reaches creatures who were never meant to hear it. Maybe it's time we learn to be quieter. Thank you for watching. If you found this video educational, please consider supporting marine conservation organizations working to protect critical ocean habitats. Links in the description. As always, take care of yourselves and take care of each other.